presentation. So that said, the next presentation is from Petra Ritter on the virtual brain.
Welcome to the presentation about the virtual brain simulation platform. Let me start with a question. What is the goal of most neuroscientists? What is your goal? The goal of most neuroscientists is to come closer to the mechanisms underlying brain function and dysfunction. And to this end, we use methods to observe the brain structure and function across various scales. For instance, with magnetic resonance imaging, we get information on structural and functional connectivity at the spatial scale of the whole brain. Yet what we actually want to understand is, how are the different observations connected? How are the processes and the underlying system causally related? And how do changes, for example, in local field potentials, firing rates, translate in functional connectivity or effective connectivity, and how into behavior? In brain network modeling, we use the anatomical skeletons, the structural connectomes, and we equip their nodes with dynamic models that do interact with the edges or via the edges connecting them. We then simulate the dynamics at each node, and we can infer the effective connectivity, that is the input currents that drive each brain region. The model provides the biophysical relationships between structure and dynamics in the brain. A platform to construct and simulate such brain network models is the virtual brain simulation platform. The virtual brain software is an open source framework, and it is free. You can download it from its website, virtualbrain.org. This eLife study demonstrates how using the structural connectome from healthy human subjects, we can construct brain network models. The models are being informed by functional data. In this case, these are simultaneously measured EEG and fMRI data. When we simulate the model, it generates functional imaging data, but at the same time, it generates the underlying neuronal processes that we cannot measure. Simulations are shown in the upper panel and across different timescales, ranging from a few milliseconds on the left side to several minutes on the right side. Corresponding measurements are shown in the lower panel. There are multiple snippets coming from different methods and even different species. We see firing rates, local field potentials, inhibitory and excitatory input currents, EEG, fMRI time series, and the switching of functional connectivity over time on the right side. The model and its simulations that you see also in the video on the left create the theory that puts the observations in mutual context. The observations all become parts of the same apparatus, parts that are causally related. In this study, we learn how the EEG algorithm is causally linked with pulsed inhibition, pulsed suppression of the firing of neurons, a mechanism that is fundamental for brain function. The dynamics that the brain generates are the result of complex interactions. To understand their origin and their principles, we need dynamic systems theory. And here's another example. Traveling waves can be seen in the intracranial EEG on the left side. they are another potentially important signature of how the brain integrates information across distant sites. Understanding the principles, how they are emerging, the traveling waves, how their direction and speed change, which has implications not just for the effective connectivity, but also for synchrony across the brain, which may be relevant for plasticity and learning. These causal relationships can be only studied using complex dynamical systems theory. With the models, we can simulate activity and we can analyze it as we would do with measured data. Here we gradually change a parameter value of the model, indicated by the x-axis in the right panels, the global scaling parameter. And under certain conditions, the model well captures the true brain activity of the patient. We can reproduce the functional connectivity and also the functional connectivity dynamics of the patient as shown in the left panels. We also reproduce EEG features as shown in the middle panels. And on the right, we see a metric of prediction quality. So what are now the general avenues of exploitation of these brain network models that rely on the structural connectomes and simulate functional connectivity dynamics for clinical research? We can use them to understand mechanisms. So which cascades of processes lead to a change related to a disease? For instance, to altered functional connectivity, or altered traveling waves, altered gating by inhibition, altered working memory, decision-making, etc. Second, simulation features can be used to augment statistical prediction models. We infer detailed neuronal processes by simulation. For instance, the detailed effective neuronal connectivity in the brain. And we can use for these as features for prediction or classification. Third, in silico intervention planning. We can do in silico tests of stimulation, medication, or surgery. Here I show you a study where we take into account the altered amyloid beta cascade in our simulations. Beta amyloid is increased in Alzheimer's disease. We can measure it with positron emission tomography PET. The value of intensity of amyloid PET can be used as a marker for the disease. But here in this study, we use it to alter in each single brain region the excitability parameter of the local model. And this assumption is based on cellular studies that show impaired function of inhibitory interneurons in the vicinity of amyloid beta. On the right side, we see a slowing of the simulated EEG. This is what we can observe in experimental data of Alzheimer's disease patients. The state-space diagram shows how the activity of neuronal populations depends on the context. One axis displays the activity or potential of pyramidal cells. The second axis displays the potential of inhibitory interneurons. The third axis shows the activity of the global brain network connected to the specific node. Depending on the global activity, the repertoire of pyramidal cells and interneurons changes. Now we can change step by step the control parameter J. This is a parameter that couples inhibitory interneurons to the pyramidal cells. With each increase of parameter J, we see how the dynamic repertoire of the pyramidal cells and interneurons changes. We see the system trajectories that translate in the simulated time series of local field potentials you can see on the right side. The change of coupling between pyramidal cells and interneurons due to the accumulation of amyloid beta protein explains the slowing that we observe in the local field potentials and in the EEG in our simulations and also in the real patients. This is a brand new publication. Brain simulation augments machine learning-based classification of dementia. It shows how inferring neuronal states through simulation with brain models constructed from the structural connectome of patients helps to improve disease classification in dementia. Potentially because it provides a more detailed resolution of neuronal affective connectivity compared to imaging data alone. Here on the left side, it is shown how adding the simulation inferred features to the feature space increases the precision of classification between disease groups. Interestingly, the relevant features are the simulated alpha band activities in the thalamus, occipital lobe, and in the dorsal stream. Now I demonstrate an example of virtual drug therapy. The connectome-based model can be treated in silico. Because of the excitotoxicity theory in dementia, it is plausible to give anti-excitatory drugs, such as NMDA antagonists. We implemented that in addition to the amyloid beta in our model. The drug modulates the effective connectivity within the local node. Here we can now analyze the effect the drug has on our brain network model. On the left, we see the input and output of pyramidal cells. And on the middle axis, the dose of the drug memantine. The curve shows how increasing the input to pyramidal cells also increases their output. But this curve gets more tilted with higher dose of the drug memantine. Memantine implements a buffer for excitotoxicity. This effect is also visible in the frequency spectrum. So virtual medications, a simulated slowing of activity in the Alzheimer's disease patient brains reverses. Connectome-based brain modeling has been used in several clinical domains. For instance, in stroke, simulation-based biomarkers have been shown to predict motor recovery. In epilepsy, there's a clinical trial running to use TVB as decision support for epilepsy surgery. Or in brain tumors, local parameters differ between tumor and non-tumor tissue. In traumatic brain injury, the local inhibition is altered. So how can you use these tools? You find them on eBrains, a research platform for neuroscience in Europe, developed by the Human Brain Project. The platform provides several tools, data and knowledge services, digital atlases, simulation services, brain-inspired technology, and medical data analytics platforms. Presently, eBrains has more than 5,000 users from more than 70 countries. 3/4 come from Europe, another quarter of users comes from other parts of the world. Brain network-based modeling has been made available as a service on eBrains, including all the required tools and workflows. The simulation engine, the framework for multi-scale co-simulation, processing pipelines for radiologic imaging data, and also simulation-ready data sets of patients and controls. Many skills are needed to construct and simulate connectome-based models, and also many types of data. eBrains helps to reduce the complexity. It provides well-organized and documented tools and data. For instance, the Virtual Brain Cloud Facility Hub provides access to brain simulation in the cloud that makes maximum use of the various services of eBrains by making them work together. You see the many authors of this publication. So this is really collaborative work. Since brain data is health data, it needs to get protected, and protection mechanisms have also been implemented already in these tools. On top of imaging-based connectivity information, other types of data and knowledge from eBrains can be integrated in the models. We can map pathway inventories on the brain and simulate the effects of changes of these cascades of processes, for example, in diseases. We can also map anatomical, cytological, or genetic expression information to the brain and increase the biological detail of our simulations. For example, on the left, you see receptor densities. Depending on their values, parameters in the model will be differing. The brain model becomes heterogeneously parameterized, becoming more similar to a real brain. And last but not least, the dynamic network models can become embodied by linking them to a robotics platform that is also part of eBrains. Here you see how output signals of the brain model control robotic hands that perform a popular finger movement task that has been studied extensively in real-world experiments. And the brain network model linked to robotic hands reproduces the specifics of movement observed in real life, but also delivers at the same time the theoretical explanations of the underlying principles. A very exciting recent development in brain network modeling is multi-scale co-simulation. Here, different parts of the brain are simulated at different degrees of detail. This is achieved by making different neuronal simulators that capture neuronal dynamics at different degrees of resolution interoperable. The brain network simulator, the virtual brain, is, for instance, coupled with spiking neuron simulators such as Nest, Anarchy, or Neuron. A recent multi-scale simulation study shows the usefulness of this approach for virtual deep brain stimulation in patients with Parkinson's disease. Doctors wish to predict the outcome of a stimulation and to optimize it for their patient. To this end, you need to monitor in your simulation the detail in the brain nuclei, in the basal ganglia, but also the spatially distributed dynamics in the brain cortex. So here, for example, this is activity simulated during deep brain stimulation. On the left side, we see the detailed spike trains in the basal ganglia. In the middle panel, we see cortical activations and deactivations. And on the right side, the thalamic firing rates during rest and during 3 different stimulation paradigms. And here we see now the cortical effects of the three different stimulation paradigms. The multi-scale brain network simulation approach enables for in silico optimization of the stimulus for an individual patient. For example, for the stimulation of the subthalamic nucleus with a monophasic stimulus, we see a deactivation in motor areas, and we see exclusively activations for the biphasic stimulus that goes to the subthalamic nucleus, or the stimulus that goes to the globus pallidus internus. And with this promising outlook, I would like to end my presentation. I would like to thank my team in Berlin and all the collaborators from the different collaborative projects. And I thank you very much for your attention.
Takes us to the end of our course. There is, of course, some time for questions. If anyone has any questions on Rick's talk on modularity or Petra's talk on the virtual brain, we can consider those now. We've still got some of the speakers here and myself as well. So are there any questions on Petra's presentation or Rick's work on modularity? Or any general comments about the course? We're always keen to hear any feedback or comments on how we can improve the course for future years. So if you've got any comments, don't hesitate to come up and chat with us or send us an email. We'd be more than happy to take on your feedback. Any general questions about network neuroscience or any discussion points? Nope. Yep.
Just, you are repeating the course next year?
Sorry?
Next year you will repeat?
It depends. It's not clear. It's not clear at this stage. But the question was, are we repeating the course next year? And yeah, we'll wait and see. Any other questions? Otherwise, I'd like to once again thank our speakers, our fantastic lineup of speakers for their time and for the outstanding presentations. I'd also like to thank the organisers for selecting our course for presentation once again this year. We've been running this course now for, I think, it's the 7th or 8th year, perhaps, and so it's great to have the course to have the course in person once again. Having it virtually for the last couple of years. So please join me once again in thanking our speakers, and I hope you enjoyed the course.